Okay, in this video we're going to talk about protein purification, production of protein and purification of that protein. So in the past, if we were to talk about insulin, which is what this paragraph is talking about, they're talking about how DNA recombination technology was used to introduce the insulin production into, for example, bacteria, and then we make the bacteria make a lot of insulin and we use it. In the past, we used to get isolate insulin from the pancreas of pigs, and then that insulin was given to humans and such. And then they're saying there's a lot of sacrificing of pigs and for the insulin, and that's no longer needed. Okay, so now what we do is we use bacteria, right? And we engineer the bacteria cells in a way that they express the protein of interest, and in this case make a production of insulin. But the problem is, okay, so here's my bacteria, right? And here's my bacterial DNA, and then I introduce the plasmid. Okay, I apologize for that. I introduce the plasmid that has insulin into the bacteria. The bacteria is not only gonna make the protein of interest that I want, it's gonna make its own proteins as well. So when I lyse the bacteria, right, open up the bacteria, and I have all the proteins coming out. How do I isolate my protein of interest from the bacterial protein? That is the question. So that is what this lab is talking about, how you perform protein purification. The way we do it, we use here they're talking about ion exchange chromatography. So chromatography is when you have in our case a long tube, okay, you have something running through the tube, and then you isolate, like you make sure the stuff that you don't want stay here. I mean the stuff you don't want come out, the stuff that you want stays, or vice versa. The stuff that you want come out, and the stuff that you don't want will stay in. Okay, so how do they control for that? Because in the lab what we did was we made sure we trapped the protein that we don't want, and then the protein that we wanted. If you remember, we had the column, right? And then we had our beads, I don't know how to pronounce them. We had our beads here, and then the protein of interest was the green fluorescent protein, right? We wanted it to diffuse through the beans and come and appear here in our tube. Okay, and the other protein that we didn't want, we wanted it to stay up there, and then we subjected this to UV light, and that's the only time when we were able to see the fluorescent protein. Okay, so how do you trap your protein? Okay, either in the beads or let them pass through. So there are 3 ways to do that. You either control for the charge, okay, affinity, or size. Okay, so it's 1, 2, and 3. Charge, affinity, and size. Okay, so when I say charge, remember how, I don't know why it's not letting me draw it all the way down. Remember how in our column we have beads. I'm sure I remember the professor talking about it lab. So those beads can have holes of different sizes. Okay, this looks bad. So let me try doing this again. So here's my bean, and it has different holes on it, different sizes. Okay, so the protein of interest that I want, I know it's a specific size, I know it's some sort of small size, so I want to trap it inside the bead. Okay, and so my protein of interest is small enough to get trapped, and it's mixed with bigger protein. Okay, so the bigger protein will not, so here my pink protein will be trapped inside the beads, my small protein will be trapped inside the beads, but the bigger ones, because they can't go inside the hull, why is it writing? It can't go inside the hull, it will pass through the beads and come out. Okay, so I, for example, trap it by size, or the other way I can do it is I make sure that my beads are if my protein is mostly, for example, positively charged, right? Okay, so and I want it to so I trapped my small protein, let's say I trapped my small protein, right? Then the next step is I know that with those small proteins there were other small proteins as well that came through and are inside the beads, and I don't want those other small proteins. I know that my protein of interest is positive and the other ones are, let's say, negative. Okay, so what I do is I make sure that my beads here, it doesn't want to write, okay, my beads here has a positive charge, okay, which is perfect. So when I put my small protein through, it will stick to the positively charged beads, okay, and then my negatively charged, so my protein of interest is negatively charged. I apologize for that. My protein of interest is negatively charged, that's why it's going to stick to the positively charged beads, and my other proteins that I'm not interested in is positively charged, those yellow ones. So the yellow ones, since they're positively charged, they will pass through because they're repelled by the positive charges. They just don't want to stick to the beads, and they'll just come out. Okay, so they'll just come out and do not want to stick to those beads. So I'm like, yay, I gathered most of the negatively charged proteins of interest that I want, but then I still have other small negatively charged proteins. Okay, what do I do in that case? So in that case, so sometimes you don't even have to do all three different types of chromatography. Sometimes doing just one is enough, but in some cases you have to run it through multiple different columns to isolate the protein. So the protein that I have here, I'm just making it a bit complicated. First I isolated it by size, but then I realized that there are other proteins that have the same size. Then I isolated it by charge, and then I realized, yes, there are other proteins that have similar charge and size. Then my third option is, so I'm just utilizing all the three ways you can isolate your proteins. But in some cases you only need one way of isolation and you'll be fine. So the third way here, the beads themselves have let's say not an antigen site, what am I looking for? So a substrate, some sort of substrate, like stuck to it. Okay, so remember how our proteins look like? They usually have a substrate binding site. So or in case of enzymes, substrate binding site or activation site or something like that, that it will like a substrate to bind to. Okay, so I know that my protein is specific for a certain substrate, that the other proteins that have the same size and charge are not specific for that substrate. So my third type of protein that has the same size and charge, why is this not working? Yeah, my third type of protein that has same specific size and charge has a different looking substrate. This is giving me a hard time, acting weird. Okay, let me retry this. So the other protein that has the same size looks like this. My protein that I want to isolate has a substrate site that looks like this, and then here I have my substrates that will only bind to my protein of interest. Okay, so I'm gonna pass the tube that has those two proteins here. So again the pink protein will bind, because I know it's gonna bind to the substrate, the black protein can't, so it will stick to the beads on the column, right? And then I know that this protein does not, the site that it has will not bind to the substrate that I have on the beads, so it will pass through and come out. Okay, so I isolated my proteins, and I used the 3 different ways to isolate my proteins, right? Size excision, which we talked about here, charge, and affinity. And again, in the case of affinity here, they tell you specific antibodies that bind to protein of interest. So you do that. Those antibodies are technically the substrate to bind to it. Binds tightly on the column and it will not let go, right? So we talked about the 3 different types. Now you have your protein stuck on your bead, right? So we saw how you can make your protein stuck on your bead. So how do you make sure that the protein, like you introduce a final wash where, okay, so this column here, I don't know why it's not letting me draw everything. So this column here has my protein of interest. How do I make my protein of, what just happened? It closed, I'm so sorry. Oh yeah, I solved this for genetics, different subject here. And this is the one I was working with. So how do I make sure that the protein of interest I have here will come out? Here the process, the way you do it is you elute. And in this process, what you do is you change the pH by either adding an acid or base or ionic conditions. So the pH will change the ionic conditions or binding conditions, okay, which will affect the interaction between the protein here and my beads. So when I do that, the protein will come out and I will collect all my protein here in this test tube. Okay, right after you do that, let's say you added a weak acid, you have to add a weak base to neutralize it. So yes, you do the elution process, you get the protein out by introducing the acid, but make sure you also introduce your weak base so you can neutralize the acid that you added, because you don't want to denature your protein completely. You want it to retain that same shape. So you don't use strong acids that will completely denature your protein. Use an acid that will change the protein enough to make sure that it lets go of the substrate on the beads, and then you add the base to make sure that they go back to their original shape. They're in that neutral environment they're used to. Okay, and so that's what this lab talks about. And the two genes that proteins we were interested in, but we only looked at one, was the green fluorescent protein. The other one was the blue fluorescent protein. And then they tell you they have specific size. This is the size. It's like 238 amino acids, and it's around 40k daltons. And that's it. Hope this helps. Thank you.